Developmental biology. The process of sexual reproduction ensures the formation of diploid zygote, which could constitute the next generation zygote as a single cell structure by an ontogenic process. The zygote undergoes various developmental processes, resulting in multicellular embryonic organization. These phases include cleavage, gastrulation, neurulation, organogenesis, and a period of growth and histological differentiation. In spite of the fact that organisms vary in their structure, form, and mode of life, the process of embryogenesis, development, and differentiation are remarkably similar in all metazoans. The later stages of development, a fundamental uniform pattern in development can be observed. The ontogenic stages also reflect the histological development of species or phylogenetic development. Realizing the mode of formation of a young individual of the next generation has always interested human mind. There is a recorded history of human natural curiosity and sexual reproduction from very early periods. The Sasrota Samhita, a monumental Indian medical book written during second or third century, as it describes the development of a human child in the mother's womb. The earliest recorded work had been done by Aristotle. As classical work, the generation animalium is concerned with the generation of animals. It describes the production and development of many kinds of animals and has in other work, De Historia Animalium. Aristotle provides an account of hen's egg. He compared the reproductive methods of different animals and provided a classification based on that. By observing the development of hen's egg, he concluded that development always proceeds from simple, formless beginning to complex organization of the adult. For this speculative idea, he provided the name epigenesis. Through his remarkable observation speculation, Aristotle established embryology as an independent field in science. Thus today, he is regarded as the founder of the science of embryology. After a period of early Greek thinkers, this discipline once again got attention of the scientists. From the beginning of the 17th century, through the contribution made by various workers like Van Beer, Haeckel, Hartwig, Wilson, Feynman, Child, Maclean, and others, rapid advancements were being made in understanding of developmental processes in animals. Modern embryology has utilized all tools made available from other branches of science and diversified into branches such as experimental embryology, chemical embryology, comparative embryology, and descriptive embryology. Such studies are meeting the challenges of today's world through works on cloning techniques tissue cultures, stem cell researches, in vitro fertilization, organ transplantation, regeneration, tissue grafting, and other medical and non-medical fields. Gametogenesis. The process of embryonic development and sexually reproducing multicellular organisms is made possible through processes of gametogenesis and fertilization. Gametogenesis is the formation of sex cells or reproductive cells or gametes. It happens in primary sex organs called gonads. The male and female gonads, namely testes and ovary, contain primordial germ cells. These cells are responsible for production of gametes. Spermatogenesis. In the testes of vertebrates, the specialized tissue for the process of spermatogenesis are located in the seminiferous tubules. The primordial germ cells of these tubules produce cells which ultimately become sperm. Mother cells are spermatogonia. Through a growth phase, the spermatogonia get converted into primary spermatocytes. These are diploid cells. They undergo meiotic cell division. Initially, the first meiotic results in the formation of secondary spermatocytes. Through second meiosis, they form spermatids. The spermatids are replied in nature by a process of spermiogenesis or spermiotheolysis. They get differentiated into specialized cells called spermatozoa. Oogenesis. A similar process happens inside the female gonad, namely the ovary, for the production of ova. This process that happens in the primordial germ cells through stages of primary oogonia, primary oocyte, and secondary oocyte. These stages are conducted by meiotic cell divisions. Thus, the final product, namely the ovum, is a haploid female reproductive cell. Fertilization. Embryogenesis could occur only after fertilization of the ovum or egg. Fertilization provides the diploid nature to the cell. Thus, all the somatic cells of the embryo will remain diploid. Further, the process of fertilization triggers or initiates the initial stages of embryogenesis. During the process of fertilization, sperm and ovum of the same species approach and some in contact with each other. The entry of sperm initiates further changes in the egg. The haploid nuclei of the sperm and formation of a diploid zygote nucleus. This process is known as syngamy or ampeximus. Types of eggs. For embryo to develop inside a fertilized egg, nutrition is needed. The amount of food needed varies from different organisms. At normally on the duration of development, food is provided in the form of yolk. It may be fatty acid or protein yolk. It is provided by the ovary during differentiation of the egg. Due to accumulation of yolk, a maturing egg rapidly increases in size. An amphibian egg yolk occurs in the form of large granules called yolk platelets. Yolk platelets contain two main proteinaceous substances, namely phosphatine and lipofuscin. The amount of yolk is an important determining factor for further patterns in embryological stages. The amount of yolk influences cleavage and gastrulation methods. The egg can be classified based on amount and distribution of yolk. Amount of yolk and egg types. In certain animals, the developmental stages are not very elaborate. The final young one born may be very simple in structure and organization. Such conditions remain in animals like Hydra, sea urchin, and Pyxis. And placental mammals and the eggs of such organism due to brevity of the growth period, the amount of yolk is much reduced. Such eggs are said to be mild or oligolecythal. In such animal, the egg need to release young ones in a more self-supportive condition. Hence, for such egg, the amount of yolk is considerable in quantity. Such eggs with moderate amount of yolk are mesolecythal eggs. Such eggs are produced by annelid worms, mollusks, and amphibians. In some animals, the growth and differentiation of embryo is much more elaborate. The growth period is sufficiently long, hence for supporting the embryo as in development, the egg contain large quantity of yolk. Such eggs are termed as megalocecal or macrolecythal eggs. The eggs of reptiles and birds are considered as macrolecythal. Eggs are covered by a calcareous shell. It is a protective structure for laying the eggs on land. Such eggs are called cladoid eggs or land eggs. This is the structure of egg. This one is shell. This of cytoplasm, yolk, albumin, vitelline membrane, shell membrane, air space, plasma. Distribution of yolk. The pattern of cleavage and the consequent distillation process are affected by the distribution of yolk within the egg according to the pattern of dispersal of yolk. The following egg types had been identified. Homolycetal or isolycetal eggs. Eggs of this type have the yolk dispersed in the entire cytoplasm. The distribution is somewhat uniform in animals, vegetal foals, and the equatorial. Animal foal, vegetal foal, isolycetal egg. Region. In such eggs, the cleavage will be deeper and may bisect the egg. Connecting the two folds or microlecythal eggs have this nature. Telolecythal egg. Animal foal, nucleus, yolk, vegetal foal. All eggs have polarity, and polarity the eggs have an innate nature for differentiated into upper animal foal and lower vegetal foal. The polar nature is mainly due to the denser material in the cytoplasm, namely yolk. The yolk and the egg will concentrated in the vegetal foal. Cytoplasm with the nucleus will occupy the upper animal fold. The extent of vegetal fold is determined by the amount of yolk. Thus, the eggs having a polarized distribution of yolk in the cytoplasm are referred to as telolecythal eggs. Mesolecythal and macrolecythal eggs remain as telolecythal eggs. Centrolecythal eggs. Cytoplasm, shell, yolk, nucleus. An egg need not be spherical always. In invertebrate animals, oval-shaped eggs are seen. The pattern of cleavage and further gastrulation also deviate from that of the vertebrates. In insects, the eggs are oval in shape. The yolk remains in the center of the egg. However, the eggs of echinoderms are similar to that of vertebrates. Cleavage and types. Frog's egg. The process of cleavage remains one of the earliest mechanical activity in the conversion of a single-celled egg into it is initiated by the sperm during fertilization. However, in parthenogenesis and parthenogenesis, egg cleavage can be commenced without the influence of fertilization. The cleavage or cellulation happens through repeated mitotic divisions. These divisions result in cells called blastomeres. In later stages of development, the blastomeres occupy different regions and differentiate into several types of body cells. The first cleavage of frog egg was observed by Somerdam In 1738, the entire process of cleavage and frog egg was studied by Proust and Thomas in 1824. With the development of microscopes, cleavage and further studies were observed in starfishes, amphioxus, and hen's egg. From all these studies, it has become clear that all division and cleavage are mitotic. The mitotic process is very rapid, and the eggs of sea urchin division of the blastoma can be observed every 30 minutes. As the cleavage progresses, the resultant daughter cells, namely blastomeres, get reduced in size. During cleavage, there is no growth in the blastomeres. The total size and volume of the embryo remains the same. The cleavage results in a compact mass of blastoma called morula. It gets transformed into blastula, while the wall of the blastula is called the blastoderm. The central cavity is called the blastocele. The planes of cleavage, and it can be divided from different planes during cleavage depending on the position of the cleavage furrow. Planes of cleavage are named meridional cleavage. The plane of cleavage lies on animal vegetal axis. It bisects both the poles of egg. Thus the egg is divided into two equal hollows. Vertical cleavage. The cleavage furrow may lie on either side of the meridional plane. The furrow passes from animal to vegetal pole. The cleavage may be unequal in size. Equatorial cleavage. This cleavage plane bisects the egg at right angles to the main axis. It lies on the equatorial plane and divides the egg into two halves. Longitudinal cleavage. It is similar to the equatorial plane, but it lies on either side of the equator. It is also called transverse or horizontal cleavage. Influence of yolk on cleavage. Yolk is needed for embryonic development. However, the fertilized egg has to undergo all stages of development and result in a suitable young form generation. Somehow, with all the influences of shock, the developmental procedures are so adapted and modified that a well-formed embryo will result. The initial influence of yolk is felt during the process of cleavage. The amount of yolk and its distribution affect the process of cleavage according to several cleavage patterns have been recognized. Total or holoblastic cleavage. In this type, the cleavage furrow bisects the entire egg. Such a cleavage may be either equal or unequal. Equal holoblastic cleavage. In microlecital and isolecital egg cleavage leads to the formation of blastomeres of equal size, for example, in Amphioxus and placental mammals. Unequal holoblastic cleavage. In mesolecital and telolecital eggs, cleavage leads to the formation of blastomers of unequal size. Among the blastomers, there are many small-sized macromers and few large-sized macromers. Meroblastic cleavage. In this type of cleavage, furrow are restricted to the active cytoplasm found either in the animal foal, macrolecital egg, or superficially surrounding the egg centrolecital egg. Meroblastic cleavage may be of two types: discoidal cleavage. A discoidal cleavage, since the macrolecital eggs contain plenty of yolk, the cytoplasm is restricted to the narrow region in the animal pole. Hence, cleavage furrows can be formed only in the disc-like animal pole region. Such a cleavage is called discoidal meroblastic cleavage. For example, birds and reptiles. Superficial cleavage. In centrolecital eggs, the cleavage is restricted to the peripheral cytoplasm of the egg. For example, insects. Laws of cleavage. Apparently, there are several cleavage patterns. However, all cleavages follow the cleavages are governed by certain basic principles or laws. Sash laws. These laws were proposed by Sash in 1877. Number one, cells tend to divide into equal daughter cells. Two, new division plane tend to the intersect the preceding plane at right angles. Balfour's law. The speed up or rate of cleavage in any region of egg is inversely proportional to the amount of yolk it contains. Cleavage of fertilized egg in frog. In frog egg, the cleavage is holoblastic and unequal. The cleavage occurs as follows. Number one, the first cleavage plane is meridianal. Initially, a furrow appears at the animal foal. It gradually extends toward the vegetal wall of the egg. It cuts the egg through its median animal vegetal polar axis and results in two equal sized blastomers. Number two, the second cleavage furrow is again meridional. It bisects the first cleavage furrow at right angles. It is a holoblastic cleavage affecting both the blastomers of the first cleavage. It results in the formation of four blastomers. In the next stage, latitudinal furrow is formed above the horizontal furrow, nearer to the animal foal. Such a furrow is due to the effluence of yolk concentration in the vegetal pole. Latitudinal furrow uniformly affects all the blastomers. It results in the formation of eight blastomers. Four of them remaining in the vegetal pole are large. They are named as macromers, and other four blastomers remain in the vegetal pole. They are named as macromers. The macromers are smaller in size than the macromers. Number four, the four set of meridional and holoblastic. They are unequal. They divide yolkless macromers and yolk-rich macromers. These cleavages result in the production of 16 blastomers. As a result of further cleavages, a ball of several small blastomers result. A closer observation reveals that while the blastomers above the equator are small and remain as macromers, the blastomers of the vegetal fold remain progressively larger. The larger blastomers are called the macromers. This one is animal fold, macromer, blastocele, macromer, macromer. Both are same terms. Vegetal fold. This is frog blastula. At the final stages of cleavage, the embryo acquires a characteristic mild oblong shape. In this stage, it is called the morula. This is mulberry-like structure. The morula initially contains a shallow cavity called the blastocele. Gradually, the blastocele space increases into a large cavity occupying the middle of the blastula. However, the blastocele mostly remain in the animal pool region in the middle of the micromers. The blastomers gradually and arrange themselves into a true epithelium called the blastoderm. The blastoderm remains true cell thick in the animal pool. The embryo having a fluid-filled blastocele and blastoderm is called blastula. It has been reported that around 12 cleavage, the blastula possesses about 4096 cells. Blastula moves to next stage, namely gastrulation, a stage in which it has about 20,000 cells. The ultimate blastula is a ball of blastomers which have formed different embryonic body layers and organs of the body. The fate of each and every blastomer has been observed and marked. A map showing various organs forming areas on the blastula is called the fate map. This map shows prospective ectoderm, mesoderm, endoderm areas. It also shows the zone of involution and zone of invagination. For the next stage of gastrulation, gastrulation in frog embryo. The process of gastrulation is a continuous activity succeeding cleavage. During this process, the blastodermal cells begin to move. They wander and occupy their prospective organ forming zone. During this movement, at one region on the blastula, the cells wander inside and occupy the cavity. At a specific below the equator, the blastoderm cells assume an elongated bottle-like shape. They move toward the interior of the blastula. As the cells move further inside, an invagination happens. A deepening of the invagination results in a cavity called the archenteron or gastrocele. The opening of the archenteron on the surface of the blastula is called blastopore. The blastopore gradually assumes a crescent-shaped shape. Finally, it becomes circular. The region dorsal to the blastoporal opening is called the dorsal lip. The lower edge may be called the ventral lip. The surface cells, representing several prospective zones of the embryo, begins to wander inside through the blastopore. These wandering of cells is termed as involution, as blastoderm, blastocele, invagination of macromers, dorsal lip of blastopore, invagination of endodermal cells. Initially, the first pharyngeal endodermal cells undergo invagination over the dorsal blastoporal lip. These cells move to the interior. They are followed by other cells. The wandering cells gradually occupy the region of the blastocele. Thus, the blastocelic cavity gets reduced. A new cavity among the involuted cells results. It is called gastrocele. The gastrocele later becomes the archenteron. The interior region of the archenteron transforms into pharyngeal region. This region remains as the foregut. The mesodermal and endodermal cells gradually occupy their positions. The inward movement of the exterior cell through the blastoporal region is called involution. The involution results in the positioning of proto-mesodermal cells and pharyngeal endodermal cells. Dorsal lip, ectoderm, endoderm, blastopore, mesoderm, yolk plug, gastrocele, this one cavity and this one cavity. Macromers, large in size. Blastocele, micromers, small in size. The mesodermal cells occupy the region between inner endodermal and outer ectodermal cells, while the exterior chord of mesodermal cell involute inside their space is taken up by the ectoderm. The expansion of the ectoderm is due to epiboly. Epiboly causes overlapping or the looping over of the gastrula by ectoderm. The blastopore is gradually covered by certain endoderm cells. The closing cells of the blastopore constitute the yolk plug. Gradually, the yolk plug withdraws to the interior and the blastopore gets reduced into narrow slit. The process of gastrulation converts the blastula into spherical, bilaterally symmetrical triploblastic gastrula. Gradually, the gastrula undergoes the process of tubulation and neurulation to become neurula. This one is pet map of progblastula, sucker, lens, ear, neural flat ectoderm, notochord, somatic mesoderm, lateral flat mesoderm, tail mesoderm. Neurula. Neurulation. Brain, pharynx, liver, yolk, notochord, neural canal, anus, digestive tract, coelom or body cavity. The process of neurulation as the formation of a neural tube. However, during this process, mesoderm and endoderm also undergo differentiation. During neurulation, the embryo lengthens along the anterior posterior axis. The dorsal side of the gastrula is lined by ectodermal cells. The presumptive area of the nervous system gets differentiated from the rest of ectoderm. It remains as medullary plate or neural plate. The neural plate later thickens and it gets raised above the general level as ridges called neural folds. In the middle of the neural fold, neural groove appears. The neural groove deepens inside the neural folds above the groove. The neural gets converted into neural tube. This tube gets detached from the surface. The neural tube remains as the prospective nervous system. The embryo at this stage is called neurula. During neurulation, the tubulation of chorda mesoderm and tubulation of endoderm also happens. This one as neural fold, neural plate, neurophore. The post-neural development of frog involves the formation of body organs. Organogenesis of frog. The primary organ rudiments from ectoderm, mesoderm, endoderm get well established during the process of gastrulation and neurulation. In the next stage, the primary organ rudiments subdivide into secondary organ rudiments. These rudiments get differentiated into various organs and organ systems. The development of ectodermal organs. The neurula of frog has three kinds of ectodermal tissues, namely epidermal ectoderm, neural ectoderm, and neural crest cells. Epidermal ectoderm. The epidermal derivatives are the skin, olfactory sense organs, ear, lateral line sense organs, median fins, external gills, lining of mouth and anus. Neural ectoderm. This layer of cells form the central nervous system and peripheral nervous system. Development of eye. The eye is photoreceptor. It is an ectodermal derivative. Its development begins even at the gastrulation stage. However, the first sign of eye formation appears when up to optic vesicle from the lateral walls of the embryonic diencephalon. Formation of optic cup. The eye develops as two lateral grows up the pro- frozen cephalon called optic vesicle. The cavity of the optic vesicle is called up to seal of the optic vesicle where the brain becomes a narrow stalk-like structure called optic stalk. The development of mesodermal organs. The mesodermal derivatives are the limbs, endoskeleton, heart, blood vessels, kidneys, coelom, and reproductive organs. Development of heart and frog. The heart is the first organ functioning because it is to circulate blood which brings nourishment from the endodermal cells to the other parts of the embryo. The initial development of heart is similar in all vertebrates. The first rudiment of heart develops after neurulation in the region beneath the pharynx from the mesenchyme and mesoderm. The heart of frog develops at the ventral side of pharynx. It is formed by mesenchyme cells. Mesenchyme cells accumulate at the mid-ventral line of pharynx and form a longitudinal strand of tissue called the rudiment of endocardium. Soon the endocardial cells get arranged and form up a thin walled tube, the lumen of which later develops into cavity of the heart. While the endocardial tube is being formed, the mesoderm of both sides unite above and under the endocardial tube, and a cavity called pericardial cilium is formed around the latter. The visceral layer of mesoderm envelopes the endocardial tube and gives rise muscular walls or myocardium, and also adds close fitting covering called the visceral pericardium. The development of endodermal organs. The predominant endodermal organs are the organs of the alimentary canal, lungs, pancreas, and urinary bladder.